Okay. Good afternoon, everyone. As this introduction, my name is Yun-Ching, also goes by Wendy from Wild-derived Lab in University of Georgia. And this afternoon, I would like to share part of my research. We found ValSten1 can be new source to peanut stem rot, which confirmed by two evaluation methods in this study. Okay. So there are many pathogens can affect peanut and cause yield loss. And my research focused on stem rot, also known as white mold in Southeast United States. The causal pathogen, The causal pathogen is Athelia rolfsii, and the endomorph name is Sclerotium rolfsii. So you can see the pathogen sign near the soil line shows on the left-hand side picture, the white fluffy mycelium, what are shown here. And the initiation of the sclerotia present on peanut pods as well. The sclerotia is the overwinter structure, which can make the management more difficult. The pie chart shows that stem rot is the most damaged disease, which causes than 40% loss annually in Georgia alone. Chemical control is a major management strategy, so farmers rely heavily on this method, and cultural practice such as the lower plant density or the earlier plant intake can also be utilized. Also, resistance variety is an option as well. So far, we don't have many resistant cultivars. Commercial cultivars show moderate to low resistance. So based on the peanut RX here, Rounded type peanut Georgia 12Y and Virginia type Bailey are resistant to stem rot relatively. On the contrast, Georgia 06G, Georgia 09 shows here are susceptible to the disease. However, it's really hard to do the resistance breeding with a narrow genetic base in peanut. Narrow genetic base means low genetic variation, so researchers actually don't have much genetic source to breed with. But while peanut can serve as a source in breeding, there are some successful examples for wild species utilization. Shows here, Bailey in United States, GPBD4 in India, BRS425 in Brazil has the leaf spot resistance, and all these resistance came from the wild peanut species. So far, wild resistance to stem rot is not utilized to cultivated peanuts. So how to breed with wild peanuts? Firstly, I would like to talk about the origin of the modern peanuts. Cultivated peanuts from a cross of two diploid wild species and followed by a chromosome doubly event. After the domestication process, it became cultivated peanut we consume now. Similar procedure was executed in lab. Two wild cross and the sterile hybrid is generated. Colchicine is applied to double the chromosome, and peanut we generated here, we call it induced allotetraploid. And these induced allotetraploid are compatible with cultivated peanuts. So my hypothesis is that wild peanut have new genetic source that can be used to improve peanut stem rot resistance in cultivated peanuts. And my objective is to evaluate the peanut stem rot resistance among induced allotetraploid by using greenhouse and field evaluation methods. In this study, 12 wild are used, and there are 13 tetraploid were used by procedure I mentioned before. Besides the tetraploid, two controls were included in the study, the 12Y as resistant and the 09B as susceptible. Greenhouse and the field evaluations were conducted in this study. There are some pros and cons for each method. So for example, greenhouse bioassay is a rapid screening method, and we can do more replication within a year, and it's a labor-saving method. On the other hand, the advantage of field is that disease is evaluated on whole plant instead of the stem cutting only in greenhouse. So in greenhouse bioassay, plantains were grown in pot, and the lateral branch was cut and put in a cup filled with potting mix. The inoculum shows here it's a mycelial plug, and we inoculate on the base of the stem near the soil line. And all the cuttings are put in a mixed chamber for high humidity. I use caliper to measure the length of the lesion on stem, and the evaluation were taken at 3, 5, 7, and 9 days after inoculation. The AUDPC was calculated based on these 4 evaluations. And we did 3 greenhouse evaluations for this study. A little bit illustration of the AUDPC. AUDPC means the area under disease progress curve. Actually, we use the formula to calculate the AUDPC. But basically, the idea of AUDPC is estimate the disease progress over time. So in this case, we have 4 evaluations here, and AUDPC sum up the area under the disease curve as S1, S2, and S3 for the first time period T1, T2, and the T3, etc. Again, 2 methods were used to evaluate the disease among allotetraploids. So rather than greenhouse, we did field evaluation as well. Actually, we didn't use the standard field evaluation. So the picture here shows the standard field evaluation. Normally, same genotypes planted as a row, which I pointed here, and pathogen is inoculated. So flag here indicate each row has the 3 inoculation sites. However, this technique is not really suitable for tetraploid, since the plant architecture of cultivated and wild are way more different, and this directly influences the disease since stem rot favor humid condition. More details about the plant architecture, 2 wild shows here on the left, and the hybrid, the cultivated peanut shows here. So you can see that the growth habit of tetraploid is in between of the wild and the cultivated peanuts. And these 2 pictures also show another 2 tetraploid growth habit as an example. So you can imagine by using the standard field evaluation I illustrated before, disease pressure is way more stronger when plant density is high, since more moisture is captured near the crown area on peanut plant. In order to generate same microclimate condition for all genotypes we tested, field evaluation was adjusted in this study. So here, 12Y was planted as background peanuts, and tested genotype were transplanted next to the background. Plants were inoculated on the base of the stem near the soil line, and this background peanut create the same humid condition for all genotypes. The procedure of this study, tetraploid and the background peanut 12Y were germinated and planted in the field at the same time, and about 2 weeks later, all tetraploids were transplanted next to the background peanut, and pathogen was inoculated individually in the field on the base of the stem. So this picture illustrates the real situation in the field. So the red arrow pointed the tested genotype, and this shows the inoculum. A 0 to 5 rating scale was used. So 0 means no disease, and 5 means the plant already died, and 1 to 4 represent in between. This rating scale was created in 2019 when we did our first trial in Tifton, Georgia. So more illustration about the rating scale, you can see that actually we just checked the lesion on main stem and lateral stem, which represent the rating scale here, and the right-hand side picture shows the death plant. The result of the greenhouse bioassay, so this graph is the AUDPC. Here, left 2 shows the susceptible O9B and resistant Y in red. And this 4 allotetraploid shows significantly smaller AUDPC value than the susceptible O9B here. Also, this 4 shows smaller AUDPC than resistant 12Y, and they are resistant as 12Y here. As I mentioned before, AUDPC represent the disease progress over time. So this graph shows the resistance for tetraploid progress. Blue line means the O9B, and the bold red line means the 12Y, and the below 4 lines represent the 4 tetraploid. So the lines below here means that these 4 genotypes continuously show smaller lesion on stem over time, and most of them are significantly lower than 09B and numerically lower than 12Y. Go to the field evaluation result. We did not include all 13 tetraploid as we did in the greenhouse since the field capacity is not enough for the 13 tetraploid. But again, left 2 shows in red shows the susceptible 09B and the resistant 12Y. And ValSten shows lowest disease among these 7 tetraploid. And the ValSten is significantly lower than 09B and as resistant as 12Y. In conclusion, allotetraploid have new resistant source to peanut stem rot. And in greenhouse bioassay, 4 tetraploid shows resistance. And among these 4, ValSten1 shows resistance in field evaluation as well. And just a quick recall, this morning, Izu talked about the ValSten1 could be the potential source to TSWV. And in my study, we found that ValSten1 also can be the new resistant source to peanut stem rot. The next step of this study, we focus on ValSten1 now. Very luckily, I can utilize 2 ValSten-derived populations, which created by our previous postdoc. Now, I'm doing the evaluation to identify resistance individually among these 2 populations. And actually, the experiment is under progress now. After I have the result, next step will be the resistance pyramiding. Plan with resistance will be crossed with 12Y and the yolk in order to get higher or more durable resistant genotypes. Last, I would like to acknowledge my advisors, committee members, and lab members in Wild Peanut Lab. Also, I would like to special thanks all people from Branniman's lab and Osasiakin's lab for their big help of field evaluations. And after that, thank you for your attention, and I would like to take any questions.
Thank you, Wendy. We've got time for questions.
Okay.
I've got a question for you, Wendy, and that's that there's a significant effort going on looking for diversity within the Athelia rolfsii or Sclerotium rolfsii within the species and see if there's differences. We've seen differences in disease control across the state that may possibly be explained by differences in the genetic populations. If we do find there's diversity amongst the Sclerotium rolfsii isolates, would you expect that to, how would you expect that to impact your breeding efforts, or would it impact it at all?
Okay, thanks for the questions. And it's a really good question. Actually, there has a paper shows the 2 isolate Sclerotium rolfsii collected in the same area show different virulence on same peanut genotypes. But actually, I have a similar research in my project. We also collect different isolate in Georgia and try to understand the diversity of the pathogen. So part of the research is that firstly, I identify the population structure, and I will choose some isolate and re-inoculate back to the peanut here. So to make sure that the peanut we breed here is really resistant to the pathogen here. But it's true that maybe the resistance shows in Georgia will not be as resistant as here shows in other states.
Okay. Thank you very much for that. Any other questions for Wendy? If not, Wendy, thank you very much for participating. Thank you for an excellent presentation, and thanks for joining us today.
Thank you.